Before 1990, developing a new medicine required navigating a collection of disparate regional requirements. Every major market operated under its own unique laws for approving drugs, often forcing companies to restart the entire regulatory process at every border. This fragmentation created severe bottlenecks. Pharmaceutical companies were forced to duplicate complex clinical trials and repeat animal testing just to satisfy regional rules, which meant life-saving treatments often faced years of delays before reaching patients. To resolve this, regulators from the US, Europe, and Japan formed the International Council for Harmonization, or ICH, in 1990. As we can see on this structural overview, they engineered a unified framework built on 4 distinct pillars: quality, safety, efficacy, and multidisciplinary guidelines. By standardizing these requirements, the ICH turned drug development from an isolated local effort into a synchronized global science. The first of these pillars is the Q series, representing quality. This mandate ensures that before any medicine reaches a pharmacy, its physical and chemical composition consistently meets strict, validated specifications. It dictates the exact parameters for everything from Q1 stability testing, proving a drug won't degrade on the shelf, to Q7, which enforces good manufacturing practices for the active ingredients. Guideline Q8 introduces the concept of quality by design. As this flowchart maps out, the variables of a manufacturing process are tested, optimized, and securely locked in long before mass production begins. Instead of reacting to batch failures at the end of the line, this forces companies to build proactive, scientifically designed manufacturing processes from the start. Without these strict Q-series parameters, replicating a complex chemical compound reliably across thousands of global facilities would be mathematically impossible. The second pillar is the S-series, focused on safety. This is the rigorous preclinical filter designed to identify toxicological risks before a drug ever comes near a human subject. It mandates critical early-stage evaluations, specifically S1 for carcinogenicity studies and S2 for genotoxicity, to detect potential DNA mutations at the cellular level. The S-series also prescribes safety protocols for higher-risk interventions, like S9, which sets the rules for the non-clinical evaluation of powerful anti-cancer drugs. By enforcing these toxicological thresholds, the S-series acts as a vital biological filter, ensuring that molecules with high toxic potential are identified and excluded before they reach human volunteers. Once human trials begin, the E-series takes over. These efficacy guidelines provide the scientific framework for all clinical studies and ongoing pharmacovigilance. The anchor of this pillar is E6, known as good clinical practice. This is the global rulebook for conducting human medical trials. As we see here, thousands of raw clinical data points and adverse event reports must converge into verified, locked structures, ensuring that trial outcomes remain resistant to manipulation. The E-guidelines further mandate that this data accounts for diverse human populations, requiring specific studies for geriatric and pediatric demographics. Because of the E-series, a medicine's promised benefits are supported by statistically significant, globally recognized human data. Finally, we have the M-series for multidisciplinary guidelines. Think of this as the administrative framework that packages all that quality, safety, and efficacy data for international review. Returning to our visual overview, in the bottom right we find M4 and M8, which dictate the electronic common technical document format. This pillar also includes M1, establishing a standardized medical terminology called MEDRA, so regulators in different countries all interpret the safety data in the exact same way. The M-series translates complex local science into a universally accepted digital format, reducing administrative friction and the risk of regulatory rejection. To see this global blueprint in action, we can track the life cycle of a new injectable drug moving through the modern ICH process. Quality guidelines lock down the impurity controls, while safety guidelines clear the biological toxicology in the lab. Next, efficacy protocols ensure the human trial data is pristine, and the multidisciplinary standards format the final global submission for regulators. For pharmaceutical companies, mastering this sequence results in fewer product recalls and fewer trial delays. For patients, it means life-saving medicines arrive more quickly and consistently. The ICH guidelines are the primary framework for global pharmaceutical regulation. By utilizing targeted compliance training to master this blueprint, Organizations ensure their medicines remain safe, effective, and globally consistent worldwide.